live, and I'm hoping that people can hear me. Right, let me get it up so I can actually see that it has gone live as well, and then we'll go from there. Right, hold on. There we go. Why is that showing it like that? Is that better? Ah, there we go. Hi, Alex. You're on there. Right, okay, let's have a look and see if I can get you a question. So, what have we got? Let's just go for a quick one. So you wanted a question. Let's go for this one then. Okay, so what have we got here? We have got a IAA. So we've got oxen. My brain is not functioning. Hi, Christine. Do you do group tuition or one-to-ones? I do both. So I have a group tuition for OCR on a Monday tonight for year 13. For year 12 it's on a Wednesday. Excuse me. And my one-to-ones are getting pretty full at the moment, so there are during the daytime I've got about 4 slots left, but it depends whether your timetable allows that. So the volume is low, you're saying. Give me 1 second. The volume is low on this. I wonder why the volume is so low. Is it because I'm so far away? Let me see if I can put this up. Let's put you closer to me. Does that sound a bit better? Is that any better? Oh, you listen on here. But message on YouTube. Right, okay, thank you. Right, let's get going on this one then. So what have we... That is better. Thank you, Alex. What have we been told? Some students investigated the effects of IAA. So you're told that's oxen. If you are studying AQA, then you need to know it as IAA. Whereas for OCR, you just need to know it as oxen concentration on the growth of roots and shoots in mustard seeds. So whenever you look at a seed, what's really important, if I have a seed, inside the seed, I have the part of it which is going to come out and grow up to be the shoot, and the other part that's going to grow down to be the root. Will you be doing year 1 content or year 2? Right now, I'm doing year 2 content because Alex has asked me to do the plant response, but I'm happy to answer any questions. I wasn't actually planning to go proper full on tonight, but here I am, as I always do. So shoot, and this is going to become the root. So when a seed germinates, when it's germinating, a part of it is going to grow up to be the shoot, and the other part is going to grow down to be the root. That looks so grainy. I really need to work out what's going on and why it's not showing it as well on this. It's really not nice. Apologies, guys. I don't know why it's doing that. But anyway, it's something I need to work at. I need to figure out what's going on with my OBS, because it wasn't doing that before, but for some reason it is doing it now. Yeah, it looks really grainy. I don't know why. But anyway, back to this. So what did they do? So interestingly, they used 8 different concentrations of IAA. So the first thing I'd be saying, right, if they've got 8 different concentrations of IAA, did they do a serial dilution for that? So straight away, I'm looking at this going, right, we've got a serial dilution because we've got 8 different concentrations. Now, they've then placed a bit of filter paper on a bed of cotton wool in the Petri dish. They've soaked it with the IAA solution. Now, if I were to evaluate this question, and I know that's not what you want, Alex, but I think it's something to consider because they're going more down the practical. Now, this is 2023's question, and there is no way that they're going to ask the same thing, but they could do something very similar. So what is it that they did? They've said they've placed the filter paper on a bed of cotton wool in the Petri dish, and they've soaked it with the IAA. How long did they soak it for? And the reason I'm asking that question, because if they soaked it for different amounts of time, would that affect the results that you've got? Maybe. They've then placed 10 seeds in a horizontal line on the damp filter paper. They've covered with a lid that has a grid marked on it as shown in the diagram. They've repeated the process with each of the different IAA. They've added distilled water to one Petri dish in place of the solution. So the fact that they've done that. Have you ever marked an AQA 25 marker? Yes, I have, but a long time ago when I used to actually teach the AQA specification more. I obviously do the marking of it for my one-to-one students, but I haven't done it officially for the exam board in a long time. That's a control. But yes, I have done the 25 markers. Place the Petri dish vertically in a warm, dark place for 2 to 3 days until the seeds have germinated. So the germination is the fact that they have now got the shoot and the root coming out, and the root and shoot have started to grow. There you go. You've been told that it's germinating in that case. Now, it's an interesting one because germination is the root and the shoot starting to grow, and then what you would have after that would be post-germination growth. What tips are there to get highest marks without out of spec marks? So if you are doing AQA and you're trying to get the maximum marks, you need to bring in new additional bits of information than just memorizing what's on the specification. So that's where for you it's harder if you've only learnt the content that's in front of you. They want you to take it a step further. They also want you to link. You need to have at least 4 topic areas linked together whenever you are doing the 25 marker. Tips for OCR A, it's very much you have to learn the content. There is a lot of content to learn. You have to learn the content. But if you learn the content, what you should do is make sure you're able to read the questions properly. A lot of students lose marking points because they're not able to see the hints that are in the exam paper. And because they can't see the hints that are there and they don't read the question, they jump. Everybody wants to jump to that end point. Everybody wants to be able to answer the question by basically just reading the question and answering it as best they can, as quick as they can, to zoom through the paper, because they feel like that way they are getting, they've not got enough time, or they're stressing about it. I'm not entirely sure what it is that's holding them back. But if you take your time in reading the question, if you take your time in giving your brain the opportunity to process what you're reading, you'll actually write your answers more formulated and with more detail than if you just rush through the paper. It's one of those things that I would really say holds the students back. So many of my students will jump to the question, and in a sense, I get it, because people will be like, oh, come on, Christine, just get on with it. And actually, that's not going to help you. By taking the time, by going through these types of questions and looking at where it could go, by priming your brain, you're actually helping yourself. So it's about taking the time. As an AQA student for my essay, it's a good idea to read through the OCR spec to find out of spec info. So yeah, I would say it's an interesting one. So tonight I have an AQA student who comes to my group tuition sessions. And so whenever the content is overlapping, which is 75% of it that's overlapping, when they come, I say to them, for example, tonight we were doing glucose concentration. The part about how the beta cell actually secretes the insulin is not part of your specification. However, it is something you could add in, which would help you potentially, if the question's on that, take you a step further. So things like that, yes, are good, but don't go too much into it. So obviously with OCR A, they've got more content to learn than AQA. So I wouldn't advise you to go into as much, so that you don't have to do, for example, biotechnology. However, they do. So it's about going into those extra bits that are relevant for your content. So manipulating genome, absolutely, 100%. That's part of your specification. Use that. The plant response is part of it. Use it. but only the IAA and the gibberellin side of things. Could you do questions that help with DNA profile and sequencing, or do you already have it on your YouTube? It's so hard. So yes, I do already have DNA profile and sequencing on my YouTube channel already. So if you go to manipulating genome playlist, which is 6.1 point, I can't think of the top of my head, 6.1.3, I want to, no, that's not right. It's not 6.1.3. What is it? Oh, you can tell my brain is not engaged in the topic area because I can't do it off the top of my head. I've been at a soft play all day, and then gymnastics, and then I was right. 6.1.3. I was right. So yeah, if you go to my playlist 6.1.3, then you'll find the videos there. You are very welcome. Hi, Ruby. How are you? You are very welcome. Very welcome, guys. Right, back to this question, back to see what else I can come out with for this part. Right, where did we get to? We're going to measure the length. So we're measuring the length of the shoot and the root by counting the number of grid squares the shoot or root covers. Yeah, back to this for you, Alex. Back to you. Okay, so why did they put the Petri dish vertically in the dark? So this is the first question. Why vertically? Why would you want to put it vertically in the dark? Oh, I'm really pleased to hear that, Ruby. I must catch up with you. So yeah, so why would we put it in the dark? What's the point in putting it in the dark and vertically? Well, it's in the dark because we want to mimic what it would be like in the soil. Because if you have a seed, what you're going to do with that seed is you're going to place that seed in the ground, and you're going to place it, normally you would pot it into soil and you place your seed in the soil. So if I want to mimic the natural environment, I would therefore want to put it in the dark. Now, the other thing we need to know is, obviously, this is going to be looking at gravitropism. Gravitropism. Now, I know that my spelling is atrocious. I know it takes me time to make sure that I am actually writing the correct words, and I write so quickly that my handwriting is a mess. If you know you do this as a student, I would advise you to make sure that you are actually doing what is needed in the exam for the examiner, because if you're making it hard for me to read your answer, I am not going to work for you. I am not going to spend more time than what is needed to try and decipher your writing. So I know that my writing is atrocious. So I know if I was going to be doing the exam, I would be slowing it down. I would be trying to make my writing a bit neater. I don't worry about this when I'm doing this with you guys. I'm doing it really quick. So one thing I would say to you though, and I say this to my students all the time, because whenever they do practice papers for me and I mark them for them, if I can't read the writing, they're not getting the marks. And so that is what will happen with your examiners. They will be like, I can't read your writing. I'm not going to read it. See you later. Obviously, not to that extent. They will try. However, what they will do is if they can't read it, they will flag it up to the next person, but you are making us work for it, and it's not that much money that they get paid. That's why I don't do it anymore, because the CPD is not there for me. When I did do it, it was really good because it meant that I was getting the experience, the guidance to think like an examiner, and I could then train my staff to be able to think like examiners and train my students. But after you've done it for a set number of years, it doesn't change. So you don't actually gain any more skills. So now the thought of actually coming back to marking again is like, nah, I'm good. I'm good. Although I do it when I review the papers for students after they've actually got their grades back. I only do a few of them because I don't gain very much from it. So it doesn't help me anymore. But it's definitely something I would say for you guys. to take your time if you know your writing is atrocious. Right. Gravitropism. We know this question is going to be asking about gravitropism because they're talking about germination. We're talking about roots and shoots. We know that gravitropism could be positive or it could be negative. Now, it's important to note that any type of tropism, that directional growth, is going to be either towards or away. So if we talk about the roots, we have positive gravitropism. If we have the shoots, we have negative gravitropism. Well, if we had light, then we'd be talking about phototropism. So we want to make sure that what we're actually assessing is tropism, the gravitropism, not phototropism, which is to do with the direction of light and that growth towards or away from the light. Okay, so what else would they say? Right, explain the purpose of the Petri dish containing distilled water. Well, we've already talked about that. I already said it acts as a control. So as I said to you before, by taking my time through this, I'm priming my brain into thinking about what might be the answers that they're looking for. So I am using it as a control. Why do you want the control? I know, Alex, I'm not showing the question. That's the reason. I'm showing you the bits that you need to know and not all of the questions because one, I can't be bothered to put it across. And also I want to keep the questions available for obviously the staff who want to use them. How can you hit the marks for AQA without writing loads? Okay, so that's an easy answer to that one because I hate writing loads and I'm lazy. I'm really selling myself here. It's great. But what I would say to you is I'm very specific in what I do write and the words that I put down. So if you are trying to hit the marks for the 25 marker, and you don't want to write loads because you don't want to write an essay, then what you need to do is you need to bullet point, and your bullet points need to be specific with the detail that is necessary to get you the marks. So what I mean by that is a lot of students think that by saying words like photosynthesis or phototropism or gravitropism, going to this question, that they've given me the mark, the word that's needed to get the mark, and actually they haven't. They've just used a big word. So you need to be able to state things like positive gravitropism is towards gravity, negative gravitropism is away from gravity. So it's about expanding on your answers and making sure that the detail that you're putting in there is to a high enough level. It's not about the filler words. It's not about the if, the ands, the buts, the ums. It's not about that. It's about the keywords and the logical connections between them would be what I would say I'd be working on if I was trying to get the highest marks for that 25 marker. Absolutely. Okay, let's have a look at another example of I'm trying to think of where let's look at this. 1 second. See, I want hard questions, but I also want them to be showing you the practical side of things. I feel like that's more what comes up. Whenever it comes to the plants, it is about the practical side of things. It's about how you can apply that. I think I've covered all the content for the plant hormones, but I haven't done exam questions yet. Okay, so if you haven't done the exam questions, then it's just about getting on and doing them. I mean, if I am always struggling to find the time to do the questions, then I'm not attempting to answer the questions. So therefore, I am holding myself back. It's one of the things I say all the time is that a lot of students, they are afraid to answer the questions. They just want to be told the answers, and they think that by listening to me talk about the answers, they're going to be able to then regurgitate that in the exam themselves. And it's not. What is important is that you practice yourself answering the questions, and you then use that to guide you on how to improve. I was told as part of feedback from my mock that I should work on PAG skills more in terms of theory questions. But you've passed your practical endorsement. So I would say the PAG skills, what they're actually saying to you, okay, is, I would agree, that's what it's more about. It's more about you working through the module 1 side of things. So by doing your PAG skills, as they've said, what they're actually asking you to do is to think about the skills that you've learned in the process of doing your PAG. So what have you learned? You've learned how to use a microscope. You've learned how to do a dissection. You've learned how to do a TLC, comes up every year. You might not have learned how to do electrophoresis. However, it's that one or that one. So they tend to say one or the other. You have learned how to do serodilution. You have learned how to draw a table of results. You have learned how to draw graphs. Interestingly, with regards to the graphs, I feel like you're potentially going to get a histogram because they haven't done a histogram in a while. What else are you assessing? Enzymes, so gas collection. The microorganisms. I do feel like your aseptic technique is coming up in more detail. Wow, that really is shocking writing, Christine. Aseptic technique. Yeah, but not in the way that you think it is. So it's important to note when you're doing this that you are aware in the questions that they will ask you won't be as simple as a nice little question that says, oh, why is it that you put the inoculating loop through the flames? It's not going to be that simple. That's GCSE level. It's going to be things like, why did they have to wait before they could put the inoculating loop into the culture broth? And so in that case, you're talking about, obviously you don't want to kill your bacteria that are in the broth. How do you recommend approaching suggest questions? So suggest questions are very much that. Suggest what you see, which again comes back to your PAG skills. So when you've got a set of results, if you've got a graph or you have got a table and you are looking at those results, can you actually give the science to explain what you're seeing? So can you draw a conclusion from those results? Can you suggest? So what they're saying to you, can you suggest what has caused the results that you've got? Now, causation and correlation obviously do not go hand in hand. If I've got a correlation, that doesn't mean that whatever I'm investigating has caused it. However, I can use my scientific knowledge to come up with a reason for what I might be observing. And that's the thing about suggest questions. It is basically trying to figure out, what do you know? How can you apply it to the context that's in front of you? And that would be my other thing that I'd say to you. With any question that you are doing, make sure what you're actually doing is reading the information and relating your answer back to that question. Suggest why the enzymes rate increased and then decrease. That's an easy one. Suggest why. Well, I'm going to suggest it's to do with the increase in temperature. I've got an increase in kinetic energy. When I get past the optimum, I therefore have a change in the tertiary structure of the protein, and that will affect the rate, and the rate will decrease. Well, that's an easy one. Whereas if it's suggest why the validity of the results has decreased based on the information that's been given, and I'm just pulling these out my head, so they probably don't make any sense. But if it's a question like that, so suggest why the validity of the results were decreased, it'd be things like they used a piece of equipment that had a lower resolution, and therefore there was more uncertainty in their data. So again, I'm just pulling these out the air. I don't know what the question is. I've just made this up. But what I'm saying to you is, think about, well, what equipment have they used? Have they told me about the equipment? Have they told me about the sample size? Have they told me that they're looking at, I was doing a question earlier about diabetes. It was an AQA question, and they had given some drugs to some rats which were having different diets. It was on the 2022 paper, and it's a case of they have rats in a lab that they're testing. So suggest why the drugs might not be useful in human trials. And that's like, well, we're using rats, we're using humans. Well, let's think about this. Well, they're both mammals. So the fact that they're both mammals means that they are within the same taxon. If I go down my kingdom, phylum, class, order, family, genus, species. So straight away, the minute I see a question that's telling me that they're doing a trial on some form of animal, and then that needs to link it to the human trials, I know that they're not humans. They're using something that's similar. However, it isn't a human, and therefore we don't know how it will actually interact with the humans. Suggest why these insects form a colony that's vertical, and somehow we are meant to know. Why these insects form a colony that's vertical? Well, if they're forming a colony and that colony is happening in a location, the suggest would be there must be something in that location that is causing them to form a colony. So maybe that has got the right nutrients there, or maybe that has got the shelter there to protect them. It really just depends what it is that you are being asked in the information that they have given you. What's the difference between accuracy and precision? Do you know that's a horrible one? It's about respiration, like CO2. You definitely look like a biology teacher. I look like a biology teacher. Love that. What does a biology teacher look like? What does any teacher look like? I'm literally, this is me, my casual at home thing, but I look like a biology teacher because I've got glasses. Oh, I love that. The difference between accuracy and precision, we don't tend to use the word accuracy anymore. If we've got precision, we're talking about precise data. We are talking about how close we are to the true value with the data that we've collected. So if we're saying we've got our mean, that's funny. My microphone is what is giving this weird shape here. Yeah, so if we're talking about the data and we want to talk about whether it is precise, again, it could be about your method when you are cutting up a piece of potato and you're trying to make sure your surface area is correct. So is it accurate? Is it precise? These aren't really words that they tend to use anymore because there is so much ambiguity with them. So I wouldn't worry too much. So yeah, precision is to do with how close to the true value it is. That was a traumatic question I'll never understand. It was part of the surface area topic. Oh, the respiration and carbon dioxide. Well, that would make sense because that was the nutrients I was talking about. You need oxygen for aerobic respiration. So yeah, you would need that. Can you please explain respiration and the yeast practical? So yeast is a type of fungi. So that's a eukaryotic organism that is going to do anaerobic and aerobic respiration. If it does anaerobic respiration, you should know that because we're talking about yeast, they are going to be producing carbon dioxide and ethanol. So that's the alcohol fermentation. So if we're going to do respiration, it would be glycolysis. Then we've got our link reaction. Then we've got our Krebs cycle. Then we've got oxidative phosphorylation. If it is aerobic respiration. If it's anaerobic respiration, we're talking about the yeast. So we know that it is alcohol fermentation. The glycolysis then leads on to alcohol fermentation, where we first have our pyruvate converted into ethanol by decarboxylation, removal of carbon dioxide, and we'll need an enzyme for that. And then we have the reduction that's going to happen. I had to think there. The reduction from the ethanol to ethanol, and that's so we can rejuvenate the NAD so that glycolysis can continue. So if we're going to do an investigation and we are looking at that investigation, how are we going to measure the rate of respiration? We'd be looking at using a respirometer or a manometer. So respirometer is the very simple respirometer where you're connecting it to a capillary tube which has a coloured bead in it, which is going to move to indicate the rate in which the oxygen is going to be taken in, and then that would result in carbon dioxide being absorbed by some form of soda lime potentially, or if you're using a manometer, it's a connected calibrated scale that you're going to use, and you've got two tubes either side. So it just depends what they decide to give you in the example. If it's a simple respirometer or if it's a manometer, it works in the same way. You're looking at the movement of the liquid and how that would indicate the respiration rate. So it's really interesting when you do the yeast practical, because you tend to do the yeast practical in a solution. And so if we're going to do that, then potentially we'd be looking at what's the different nutrients that we provide it with. For example, are we going to give it glucose? Are we going to give it maltose? Are we going to give it lactose? Are we going to give it sucrose? Are we going to give it starch? What's the nutrients that we're giving it? And how does the rate of respiration change based on these different substances that they're being given? So it really depends. You can go so many different ways with that question. But it's about measuring the rate of oxygen uptake or measuring the rate of carbon dioxide being released. It just depends, the question. Kimmy, is it too late to start revising now? No, absolutely not. Everything you do from now until your actual exam is going to help you to get to a better place and being prepared for making the progress. It's never too late. And if you're saying, oh, there's no point, you are automatically setting yourself up for failure. Don't do it. You have plenty of time. I have seen students move up a grade, if not 2 grades, with the right dedication, with the right effort and the right focus in the right areas. 2 grades they've moved up in the space from Easter until their actual exam. So it's absolutely not too late. I want to do, oh, hold on one second. Sorry, I missed that one. Where'd it go? So many likes. Thank you so much for likes. I want to do past papers, but we haven't finished content. Should I just do AS? No, do all the papers. Even if you haven't finished the content yet, still do the past papers. And the reason I would say that, yes, I am getting your questions. With the RQ value, hold on, let me answer this one, then I'll come back to you. But yes, I can see your questions. And you are basically, everything you've said is correct. So therefore, that's why I've not really focused on that. So I apologize. But coming back to doing the past papers, the more past papers you do, even when you don't know the content, even attempting it is going to help you, because you will then start to be thinking about it. And if you don't know it, go and research it. Go and watch my YouTube videos. Go and try it, because actually, I'm a trained teacher. These videos that I've put out there aren't just anyone doing it. This is me. I have got 20 years experience of teaching. I went to Cambridge, and just because I'm not in a school setting anymore, because I'm all online, doesn't mean they're not to a high standard. I basically made these videos not just for you, but also for teachers out there, because as a head of biology, what I tend to find is the new teachers that are coming through, and I remember being a new teacher, I didn't have the knowledge. I didn't have the knowledge to be able to teach. I was still learning how to teach. So I didn't have the time to go and do the research and learn all the content. And when I moved from being a regular teacher to a head of biology, I went from teaching AQA actually, and OCR, because we did both of the A levels. I went to an IB school, and when I went to an IB school, I became the head of biologist. I'd never heard of the IB before. I started teaching the IB, and I was like, I don't know a lot of biology that I have to teach, because what my previous school had done is they'd gone, right, you're teacher A, you're teacher B, and this happens in so many schools. Teacher A is going to teach this module, teacher B is going to teach this module. And so therefore what then happened was I was really good in some of the topic areas, but I didn't have a clue about the other topic areas. And so I really struggled. And then I went to teach the IB, was head of the subject, and I was like, I need to learn this. And my teachers hated me, because what I did is I made them piggyback. So we had to do back-to-back lessons. So if there were two teachers who were teaching the same class, they had to talk to each other. They had to know where they'd got to, and therefore they had to learn the whole content. Because if a student comes to me and asks me about a different topic area that I haven't taught, I don't know the answer. Well, I do now. And with OCR A and with AQA, I am ingrained in their content. And so, yes, I've not built my resources around AQA, because you've got loads of resources out there for AQA, but there was nothing for OCR. When I came back to teaching OCR, there was nothing for me to look at, nothing for me to gain that quick recap of the content. Yes, I do see your message. If you want to send me an email, there's a link in my profile on YouTube where you can send me an email direct to my mailbox, and I can respond to that. But you have to be aware my time restraints, and it's way past my bedtime. I just noticed it's past 10 o'clock. See, this is where I'm just chatting away. I won't do the lip of Henley tonight, obviously, because it's so late, but I have got a video on the lip of Henley. That one is 5.1.3, I want to say. If it's not 5.1.3, it's 5.1.2. It's in the excretion module. It's definitely there. Oh, thank you. Yes, please do watch my YouTube videos, because I earn money if you watch my YouTube videos. Very little money, but it's exciting. I get my first payment this month. It's taken me 5 months, but I get paid from YouTube this month. I'm so excited. £76 for 5 months of you guys watching my videos. It's amazing. But yeah, please watch my videos. And so therefore I can watch. Oh, don't say no. I'm joking. BioRich is actually really good. She is very good as well. Do you recommend STEM up? I don't know about it. Enzymes and transport is everywhere. Definitely make sure yes, absolutely 100%. Definitely make sure you get that. OCR is dry. Yeah, it was a weird one. Much AQA, no. Exactly. That's why I built it. Is it enough time to pass GCSEs if I lock in with revision? Absolutely 100%. Thank you for watching my videos, guys. I really do appreciate it. I want to make more, but obviously I have to prioritize where I get my money coming in, and that's through my group tuitions and my one-to-ones. And obviously for those of you who have bought my study guides, thank you so much, because that as well is a form of income, and I'm spending a lot of time building them. I've not launched it yet, but I've just finished a chi-squared workbook, which has 40 marks worth of questions, and so it's like 53 minutes worth of you testing yourself. I'm going to do one for the t-test and one for the what's the other one, Spearman's rank as well. So I'm in the process of doing that. I've got a busy day on Wednesday and Thursday because I am teaching non-stop. I've got 7 hours, but I will be back on Wednesday evening. After I finish my group tuition, I will be back and I will do a proper session on Wednesday. So please do keep watching. Do I have a PhD? No, I don't. I have a master's from Cambridge. I have 2 postgrads from Cambridge and a master's, but I don't have a PhD. I don't have the time or the inclination for a PhD. There isn't a secret chat. No, I'm also on my TikTok as well as here. And so I'm answering the questions that are coming through. And they're coming through thick and fast on this one. Focus on the comment chapters first. Right. I am definitely going to go now. So guys, thank you for joining me. I will be back on Wednesday, so please do come and join me. In the meantime, also have a break. Wednesdays is the day that I tend to